Warm greetings to one and all present over here. Me, Aditi Sharma, and my groupmates Lata, Aditya, Kevat will be discussing about food microbiology. Food microbiology is nothing but the study of microorganisms that inhibit, create, or contaminate food. This includes the study of microorganisms causing food spoilage, pathogens that may cause disease, especially if food is improperly cooked or stored. Microbes used to produce fermented foods such as bread, cheese, yogurt, beer, and wine. Also, microbes are useful for producing probiotics. We'll be talking about pH, water activity or potential, nutrition content, accessory food substances. Interactions between microorganisms and our foods are sometimes beneficial. The interaction between microorganism, plant, and animals are natural and constant. The ecological role of microorganisms and their importance in all the geochemical cycle in nature. In most cases, microorganisms use our food supply as a source of nutrition for their own growth. This, of course, can result in deterioration of the food by increasing the number, utilizing nutrients, producing enzymatic changes, and contributing of flavors by means of breakdown of product or synthesis of new compounds. They can spoil a food. This is normal consequence of the action of microorganisms, since one of their function in nature is to convert reduced form of carbon, nitrogen, sulfur in dead plants and animals to the oxidized form required by plants, which in turn are consumed by animals. So by simply doing their thing in nature, they frequently can render our food supply unfit for consumption. To prevent this, we minimize the contact between microorganisms and our food by preventing by keeping foods in the refrigerator and also eliminate microorganisms from our food or at least adjust conditions of storage to prevent their growth preservation. Factors affecting microbial growth. Environmental factors that affects microbial growth are moisture, oxygen, pH, light, carbon dioxide, temperature, osmotic effect, mechanical and sonic stress. Hydrogen ion concentration, pH. In most pH is a term used to describe the acidity or alkalinity of a solution. A pH 7 equal amount of acid and alkali, it is neutral. pH 7 below 7 is acidic and above 7 is alkaline. It has profound effect on the growth of microorganisms. Most bacteria grow best at about pH 7 and grow poorly or not at all at below pH 4. Yeast and molds are more acid tolerant than bacteria. Every microorganism has a minimal and a maximal and an optional pH for growth. Microbial cells are significantly affected by the pH of food because they apparently have no mechanism for adjusting their internal pH. Hydrogen ion concentration pH. The pH of food varies, although most foods are neutral or acidic. Foods with low pH values usually are not readily spoiled by bacteria and are more susceptible to spoilage by yeast and molds. A food with inherently low pH will therefore tend to be more stable microbiologically than a neutral food. The excellent keeping quality of the following food is related to their restrictive pH. Fruits, soft drinks, fermented milks, and pickles. Some foods have a low pH because of inherent acidity. Others, example the fermented products, have a low pH because of developed acidity from the accumulation of a lactic acid during fermentation. The lactic acid bacteria are exception. They can grow in high acid food and produce acid to give us sour milk, pickles, fermented meats, and similar products. Most bacteria are favored by a pH near neutrality, although some, as the acid former, are favored by the moderate acidity. And other example, the actively proteolytic bacteria can grow in media with a high alkaline pH, as found in the white of a stored egg. The buffers in a food, that is the compound that resists changes in a pH, are importantly not for the buffering capacity, but also for their ability to be especially effective within a certain pH range. Buffers permit an acid or alkaline fermentation to go on longer with a greater yield of products and organisms than would otherwise be possible. Vegetable juices have low buffering power, permitting an appreciable decrease in pH with the production of only small amounts of acid by the lactic acid bacteria during the early part of sauerkraut and pickle fermentation. This enables the lactate to suppress the undesirable pactane hydrolyzing and proteolytic competing organism. Low buffering power makes for a more rapidly appearing succession of microorganisms during a fermentation than does high buffering power. Now I would like to ask Kevat.
Hydrogen ion concentration, pH. The inhibitory properties of many of the organic acids, including acetic, benzoic, citric, lactic, propanoic, and sorbic acids, make them widely used as acidulants or preservatives in food. Also, changes in titrable acidity are not always evident from pH measurements. Not only the rates of growth of microorganisms affected by pH, so are the rates of survival during storage, heating, drying, and other forms of processing. Also, the initial pH may be suitable, but because of competitive flora or growth of the organism itself, the pH may become unfavorable. Conversely, the initial pH may be restrictive, but the growth of a limited number of microorganisms may alter the pH to a range that is more favorable for the growth of the microorganisms. Organism categorized according to the pH. Acidophiles grows between pH 0 to 5.5. Examples: Ferroplasma and Thiobacillus. Alkalophiles group between pH range of 7.5 to 14. Example, Thermococcus acidophilus. Neutrophils grow between pH 5.5 to 8.0. Example, Lactobacillus acidophilus and E. coli. Bacteria prefer media of pH near neutrality and usually cannot tolerate pH values much below 4 to 5. Moisture requirement, the concept of water activity. Microorganisms have an absolute demand for water. For, for without water, no growth can occur. This water requirement is best expressed in terms of available water or water activity, AW. The water pressure, the vapor pressure of the solution divided by the vapor pressure of the solvent. The AW of pure water would be 1.00 and for an 1.0 meters metric solution of the ideal solute, the AW would be 0.9823. The AW would be in equilibrium with the relative humidity RH of the atmosphere about the food. In other words, AW into 100 equals equilibrium relative humidity ERH percentage. A relative humidity about a food corresponding to an AW lower than that of the food would tend to dry the surface of the food. Conversely, conversely, if the relative humidity were higher than the corresponding to the AW, the latter would be increased at the surface of the food. Moisture requirement, the concept of water activity. Water is made unavailable in various ways. Solute, solutes and ions ties up with water in solution. Therefore, an increase in concentration of dissolved substances such as sugar, salts, is in effect is in fact is in effect a drying of material. Not only is water tied up by solutes, but water tends to leave the microbial cells by osmosis if there is a higher concentration of solute outside the cells than inside. Hydrophilic colloids make water unavailable. As little as 3 to 4% agar in a medium may prevent bacterial growth by leaving too little available moisture. Water, water of crystallization or hydration is usually unavailable to microorganisms. Water itself, when crystallized as ice, no longer can be used by microbial cells. The AW of water ice mixture decrease with a decrease in temperature below 0 degrees. The AW values of pure water are 1.00 at 0 degrees Celsius, 0.953 at 5 degrees Celsius, 0.907 at minus 10 degrees Celsius, and so on. In a food, as more ice is formed, the concentration of solute is water in the unfrozen water is increased, lowering at AW. Thank you. Now I would like to call Aditya upon.
Thank you, Kevat. Factor affecting AW requirement. Factors that may affect AW requirement of microorganisms include the following. The kind of solute employed to reduce to AW. For many organisms, especially the molds, the lowest AW for growth is practically independent of the kind of solute used. Other organisms, however, have a low limiting AU value with some solute than with others. Potassium chloride, for example, usually is less toxic than sodium chloride and in turn is less inhibitory than sodium sulfate. The nutritive value of the culture medium. In general, the better the medium for growth, the lower the limiting AW. Temperature. Most organisms have the greatest tolerance to low AW at the optimal temperature. Oxygen supply. Growth of aerobes takes place at lower AW in the presence of air than its absence, and the reverse is true for anaerobes. pH. Most organisms are tolerant of low AW at pH value near neutrality than in acid or alkaline media. Inhibitors. The presence of inhibitors narrows the range of AW for growth of microorganisms. Method for control of AWR. The first, equilibration with controlling solution. Second, determination of water sorption isotherm for the food, given by Iglesias and Sheriff. And third, addition of solutes. Moisture requirement. A consideration of the moisture requirement of microorganism leads to some general conclusion. Each organism has its own characteristic optimal AW and its own range AW for growth for a given set of environmental conditions. Factors affecting the moisture requirement of organism are A. The nutritive properties of the substrate, its pH, its content of inhibitory substance, availability of free oxygen, and temperature. An unfavorable AW will result not only in reduction in the rate of growth but also in a lower maximal yield of seed cell. The more unfavorable the AW of the substrate, the greater the delay in initiation of growth or germination of spores. This often is an important in food preservation as is reduction in the rate of growth of organism. In general, bacteria requires more moisture than yeast and yeast more than mold, which shows lower limiting value of AW for bacteria, yeast, and mold respectively. There are notable exceptions to this generalization. However, as some molds have a higher minimal AW for growth and as that for spore germination than do many yeast and some bacteria. Lowest AW values permitting growth of spoilage organism. For bacteria, it is 0.91. For yeast, it is 0.88. For molds, it is 0.80. For halophilic, it is 0.75. For xerophilic, it is 0.65. And for osmophilic, it is 0.60. Microorganisms that can grow in high concentration of solutes, example sugar and salt, obviously have a low minimal AW. Halophilic bacteria require minimal concentration of dissolved sodium chloride for growth. Osmophilic yeast grow best in high concentration of sugar. Oxidation reduction potential. The oxygen tension or partial pressure of oxygen above the food and the OR potential of reducing and oxidizing power of the food itself influence the type of organism which grow and hence the changes produced in the food. The OR potential of the food is determined by the first, the characteristic OR potential of the original food. Second, the poisoning capacity, that is the resistance to change in potential of the food. Third, the oxygen tension of the atmosphere about the food. And fourth, the access which the atmosphere has to the food. Air has high oxygen tension, but the headspace in an evacuated can of food would have a low oxygen tension. From the standpoint of the ability to use free oxygen, microorganisms have been classified as aerobic when they require free oxygen, anaerobic when they require best but in the absence of free oxygen, and facultative when they grow well either aerobically or anaerobically. Moles are aerobic. Most yeast grow best aerobically, and bacteria of different kind may be aerobic, anaerobic, or facultative. From the standpoint of the OR potential, a high, that is oxidizing potential, favors aerobes but will permit the growth of facultative organisms, and a low reducing potential favors anaerobic or facultative organism. However, some organisms that are considered aerobic can grow, but not well, at surprisingly low OR potential. Growth of an organism may alter the OR potential of a food enough to distend their organism. Anaerobes, for example, may lower the OR potential to a level inhibitory to aerobes. Now, I would like to hand over the further presentation to Lata Chaudhary. Thank you.
Thank you so much, Aditya. The topic that I'm going to explain you all is nutrient content. Now let's understand what are the kinds and proportions. The kinds and proportions of nutrients in the foods are all important in determining what organism is most likely to grow. Consideration must be given to 1. Foods for energy, 2nd. Foods for growth, and 3rd. Accessory food substances or vitamins which may be necessary for energy or growth. Foods for energy. The carbohydrates, especially the sugars, are most commonly used as an energy source, but other carbon compounds may serve, example, esters, alcohols, peptides, amino acids, and organic acids, and also their salts. Complex carbohydrates, for example, cellulose, can be utilized by comparatively few organisms, and starch can be hydrolyzed by only a limited number of organisms. Microorganisms differ even in the ability to use some of the simpler soluble sugars. Many organisms cannot use the disaccharide lactose, that is milk sugar, and therefore do not grow well in milk. Maltose is not attacked by some yeast. Bacteria often are identified and classified on the basis of the ability or inability to utilize various sugars and alcohols. Most organisms, if they utilize sugars at all, can use glucose. The ability of microorganisms to hydrolyze pectin, which is characteristics of some kinds of bacteria and many molds, is important, of course, in the softening or rotting of fruits and vegetables or fermented products from them. Foods for growth. Microorganisms differ in their ability to use various nitrogenous compounds as a source of nitrogen for growth. Many organisms are unable to hydrolyze proteins and hence cannot get nitrogen from them without help from a proteolytic organism. One protein may be better source of nitrogenous food than other because of different products formed during hydrolysis, especially peptides and amino acids. Peptides, amino acids, urea, ammonia, and other simpler nitrogenous compounds may be available to some organisms but not to others, or may be usable under some environmental conditions but not under others. Some of the lactic acid bacteria grow best with polypeptides, has nitrogenous foods, cannot attack casein, and do not grow well with only a limited number of kinds of amino acids present. The presence of fermentable carbohydrate in a substrate usually results in an acid fermentation and suppression of proteolytic bacteria, and hence in what is called as sparing, action on the nitrogen compounds. Also, the production of obnoxious nitrogenous products is prevented or inhibited. Many kinds of molds are proteolytic, but comparatively few genera and species of bacteria and very few yeast are actively proteolytic. In general, proteolytic bacteria grow best at pH values near neutrality and are inhibited by acidity, although there are exceptions such as proteolysis by the acid proteolytic bacteria that hydrolyze protein while producing acid. Carbon for growth may come partly from carbon dioxide, but more often it comes from organic compounds. The minerals required by microorganisms are nearly always present at the low levels required, but occasionally an essential mineral may be tied up so that it is unavailable, lacking, or present in insufficient amounts. An example is milk, which contains insufficient iron for pigmentation of the spores of penicillin, group 14. Bacteria causing septicemia usually have the ability to bind some of the iron in blood. Only strains which can compete for transferring iron are able to grow well in human blood. Accessory food substances or vitamins. Some microorganisms are unable to manufacture some or all of the vitamins needed and must have them furnished. Most natural plant and animal foodstuffs contain an array of these vitamins, but some may be low in amount of or lacking. Thus, meats are high in B vitamins and fruits are low, but fruits are high in ascorbic acid. Egg white contains biotin but also contains avidin, which ties up biotin, making it unavailable to microorganisms and eliminating as possible spoilage organisms, those which must have biotin supplied. The processing of foods often reduces the vitamin content. Thiamine, pantothenic acid, the folic acid group, the ascorbic acid in air are heat liable, and drying causes a loss in vitamins such as thiamine and ascorbic acid. Even storage of foods for a long period, especially if the storage temperature is elevated, may result in a decrease in the level of some of the accessory growth factors. Thank you, everyone. Thank you, ma'am, for giving us the opportunity to present our teamwork. Thank you so much.